Hello, we're going to be presenting on a very familiar biological topic, which is DNA fingerprinting and forensics. My name is Aiden Brocious.
My name's Nick Bidwell.
And Sam Bitzer.
Okay, first a little background knowledge. We're going to first specify what DNA is, or otherwise known as deoxyribonucleic acid. Did not pronounce that right, but all right. Is what stores all of living organisms' genetic information. It was first discovered in 1869 by a man named Francis Crick. The useful aspect of DNA is that it is unique to each individual. Knowing the specifics of a strand of DNA can help you determine the exact person that it came from. This being said, DNA is very useful in aid of forensic analysis. This empowers law enforcement to be able to scientifically pinpoint a suspect or a person of interest.
So I'm going to talk about nanotechnology and forensics. The implementation of nanotechnology has enhanced the overall fingerprinting and forensics process. The quality of the fingerprints using nanotechnology has been greatly improved from the original powders used for fingerprinting. An issue with previous methods of lifting fingerprints were that some surfaces were very difficult to obtain prints off of. Nanotechnology helps solve this issue. The nanopowders used in the fingerprinting process measure under 100 nanometers, which allowed for much better clarity of the fingerprint obtained. The newest materials allow for the most detailed fingerprints to be lifted off of mostly every surface. The process of analyzing the fingerprints after they are lifted from the surface has also been sped up using nanotechnology. In the past, it took many processes to obtain a clear fingerprint, but with nanotechnology, the process was made quicker and more effective at the same time. So the environmental effects of nanomaterials. When the first set of nanomaterials were released, they were found to be toxic to the environment, and they were only able to be used in the lab. The pollution that the materials can emit are dangerous to human health as well. In order to work with these materials to further study them, the lab practices were very strict. After further studies were conducted with the different materials, they were able to be stabilized so that they could be put into use in the field. The field of forensics has greatly benefited from the use of nanotechnology.
So now we're going to talk about gold nanomaterials. Gold nanomaterials are one of the materials that are currently in use. This fine powder allows for the best quality of fingerprints to be lifted. The small particles and chemical makeup allow for the particles to mesh seamlessly together on a print so that it can be lifted clearly. This aids in the ability to lift prints off of what could be considered tricky surfaces. These powders also streamline the process by cutting out the steps of water treatments and reductions which previous methods required when pulling prints. Now we'll talk about BioMEMS. BioMEMS are an example of a nanotechnology. MEMS is an acronym for Microelectrical Mechanical Systems, and BioMEMS is referring to MEMS specifically used for biomedical applications. MEMS are made up of microsensors, microactuators, microelectronics, and microstructures to create the microsystems that are used to pull information from biological samples. An example of a BioMEMS for the use in forensic analysis is called a microfluidics chip, which is shown in the image on the right. This chip can be used to sequence DNA quickly and effectively. The way that this works is that a DNA reservoir is placed next to the chip, and it enters the chip through micropassageways which are subjected to electric fields. The different nucleotides react differently to the fields, which directs the fragments to split into the separation chamber into their specific holding spots. The interesting part about this chip is the fluid dynamics inside of the MEMS. The equation on the bottom gives the relationship taking into account the free space permittivity and the permittivity of the medium surrounding the particles radius, the Clausius-Muscatus factor, and the magnitude gradient of the electric field. This concept allows for forces to be studied at a microscopic level. Once this technical step has taken place, the fragments can be studied using computer software to determine specific DNA structure. This technology gives the ability to source biomedical information which has been obtained in small quantities in an extremely efficient manner, and it can also be completed in minutes opposed to hours, which speeds the overall process up, which is great for a forensic situation where time matters.
Alright, lastly we'll be talking about the software. Software has become the most powerful tool when analyzing DNA collected. Programs today have now been designed to evaluate the DNA by taking one strand off the double helix and then compiling together with other strands. Once the DNA strand is compiled, it is then programmed to another DNA strand to create possible matches. The possible matches are then chemically tested to confirm or discredit the match. This process essentially explains what Microsoft just put out, which is the DNA displacement tool. Using this technology, we can further research genetic diseases by computing and solving the possible nucleotide matches that cause these diseases. And in conclusion, the use of DNA fingerprinting and forensics is constantly evolving. The new techniques are always being invented, and different implementations of these types of analysis are being studied to be useful in many other fields.